Okay. Good afternoon. Can you hear me? Yes. Hello, people. Can you hear me? Okay. Good. Okay. Right. So I hope everyone can hear me. Okay. So today we are starting. We are going to get another topic in your another lesson in your syllabus out of the way. Okay, because you will see at least two MCQs and a part of the structured essay, and also definitely one third of a short note is going to be from applied. Right, one third of a short note is going to be from applied. So up to maybe so two MCQs structured essay. So 3 marks, about one third of a essay. So maybe up to 6 marks, 5 marks could be most likely 5 marks could be from applied biology. Okay, so that's why I'm going to cover this as a this not just for the sake of covering, but we are going to cover it in a way that is suitable for your time being. Then so you are right now exactly almost 30 days away from the exam. Yes, so at this point what we have to do is we have to get through the lesson, as well as we have to see how you can be questioned from that particular lesson rather than going into deep details, right? So basically we are going to go through the content in the resource. We are going to highlight everything, okay, and then we are going to take some small notes in the tute, and most importantly, we are going to discuss some likely questions, okay, as well as while having a understanding of bit difficult to understand concepts, okay, then you can get into sort of studying it, self studying it by yourself, getting into it to your memory. I'm going to help it as much as possible, so that it's easier for you to take it into your memory rather than blindly trying to memorize. We'll just go through the lesson as fast, as sensible as possible, and then it's we'll add some questions into some model paper questions into our papers as well. Okay, then that will be enough of that. Then we'll have some time to go through a bit of molecular revision again, right? If we have time. So in another announcements, we'll be starting a small guest essay program this Friday onwards. Okay, so at the end of the paper class, we'll have a small guest essay discussion. Okay, so be prepared for that as well. I'll be sending the essay topics beforehand, and you will have to write the essays before you join the paper class, and then we'll go through the marking scheme. Understood? Okay, right. So we'll get into the the applied biology. I hope all of you have got the resource book. Yes, unit 10. So I'll be referring the resource book as well. Okay, because we are going to rely on it mainly as a note. Okay. Have you all downloaded the PDF I sent you? Because I sent it in the last minute. But I hope all of you have downloaded it. Yes, hands up who didn't get the PDF of this document that I have opened here. Anyone who didn't get this in the meeting? Hands up. Everybody got it right? Okay. So take out the resource books. Okay. So if you haven't touched the applied biology section yet, okay, you can start highlighting. Okay, page number 38. Okay, so we are going to start with a topic that is called as aquaculture. Okay, so it's going to be a bit of general knowledge. So what I have to say at the beginning of this lesson is this lesson is mostly common general knowledge. Okay, with a bit of things that are, I mean, some things that are added, some things that might be out of your normal knowledge, such as stem cells and certain nanotechnologies and those kind of things. But in this early part of this lesson, you will see this aquarium maintenance. You know these things. If you had a fish tank in your home, you know this. Then there, when you come to food preservation, I mean, from practical knowledge, you know these things. What are the ways that you can preserve food? From your household knowledge, you know these things. Then certain ways of dengue mosquito eradication. You have listened to so many seminars and workshops on dengue eradication and filaria eradication. Those things you know, right? So I'm going to mainly focus on the things that you are learning for the first time, such as the symptoms of filaria, symptoms of dengue, certain stem cell techniques, and also the Human Genome Project, which is this one, Human Genome Project. I'll just go through the timeline of the Human Genome Project, which is going to make it easier for you to understand. Okay. Food and Agriculture Organization defines aquaculture as farming of aquatic organisms, including fish, mollusks, and crustaceans, and aquatic plants. Can you highlight that? That's a definition. Aquatic organisms, fish, mollusks, crustaceans, and aquatic plants. All those comes under aquaculture. Okay, aquaculture. Okay, so farming edible aquatic organisms is accepted as one of the best solutions for the increasing human population. So you talked about, we learned about the problems, I mean, solutions for the problems, issues pertaining to biology in the introduction to biology lesson. So one of the main problems is what? We are going to face a food shortage in near future, right? For that, this sort of culturing of these organisms could be a good solution. Okay. A healthy diet high in protein is necessary to ensure that, okay, so I'll have the resource book here, right? Then that makes it easier for you and me both. Another, high in protein is necessary is to ensure the growing population does not succumb to sickness and diseases due to lack of, okay. Harvest from wild sources of fish, crustaceans, and other aquatic species cannot keep up, right? So that is because the widely, I mean, in the ocean, naturally reproducing organisms are not just enough, right? If you have seen the amount of fishing ships that are in the ocean, they harvest a massive amount of tons and tons of fish and other aquatic organisms in a single day. Okay, trying to match the demand through commercial fishing would eventually result in overfishing and the loss of these species entirely, right? Therefore, aquaculture is essential to maintain the human demand for fish and fishery products. And also, it relieves the strain on wild species. So, they can ask you what are the, I mean, what are the sort of positive things or the benefits of aquaculture, right? One of the best things to write is relieves the strain on wild species so that you don't have to rely on natural reproduction of these organisms. Okay, allow them to continue to be a significant food source. If you completely eat, I mean, if you completely harvest and overfish the natural organisms in the ocean, those natural food webs and those things will be destroyed so that they will sort of cease to exist, right? So once they are exist in the natural environment, sometimes they would be lost forever. So it's good to have aquaculture as a sort of a side, let's say, it's a as a method of obtaining this sort of aquatic organisms of food. Okay, this is a past paper question. What are the general characteristics of a species that could be cultured? I think 2021 paper, short note. Okay, characteristics of a species that could be cultured. So this is general knowledge. So you have to keep in mind, if you want to grow a certain organism in captivity in artificial environments, so that you can take it as food, you can harvest it as food. What are the good characteristics of that organism, right? This is general knowledge things. Should be able to withstand the climate of the region. Okay, so the first point is climate. It should grow well in prevailing physical and chemical parameters of the water. Okay, so if you, in the water that is available to you, it should grow well in your currently available physical and chemical conditions. Okay, should be easy to breed. These, I told you, these are obvious things. So that sufficient number of fertilized eggs, early fry, should be obtained. Early fry means sometimes some aquatic organisms, they, you need the larval stage of certain, probably certain these crustaceans or prawns or whatever, you call them fry. So you sometimes even import them, different species. Okay. Techniques of incubation of fertilized eggs and rearing techniques of hatchlings, early fry should be available. Okay, so that the production of sufficient number of young organisms is easy, right? Easy incubation, easy incubation. Okay, I'm going to tell you what are the keywords that you can use to remember this sentence. Okay, if the eggs, larvae, fry, fingerlings, juveniles, and the adults of the species are hardy, it is easier for the hatchery manager farmer. What do you mean by hardy? That they are resistant. What are the things that they should be resistant to? Diseases, changes in the physical conditions, changes in the food source, right? All those things should be faced. I mean, the organism should not be affected much. The food and feeding habits of each development stage should be known. So it should be the knowledge should be widely available. Okay. It should not reproduce in grow out ponds or tanks. Right. I'm gonna make import. Right. So certain species that you use as food, they are separated according to the, usually the males and females are separated, and they are kept in what we call as grow out tanks. When we grow out tanks, right? So those grow out tanks are designed so that they only get bigger. So they are provided with food, but they should not be able to reproduce. That means there are going to be young organisms in these grow out tanks. So that's going to be a problem. So they should not reproduce in these grow out tanks. If it reaches sexual maturation relatively late, it is advantageous, right? That means they have a extended growth period. Can you understand? That means they grow and grow for a long time. I mean, not for a long time, but they have time to get bigger before they reproduce again, right? Then you are going to have young organisms again. No? But before that, if you can have a big adult, it could be a fish or a whatever, that means it's going to produce, it's, you can take a lot of food from it, right? It should accept formulated food, because we are going to feed it artificial food, no, right? So it should be able to take the formulated food. It should be an efficient converter of economical foodstuffs. So whatever the food that you give it, of course you need to have profit margin. So you spend money on the food, but their harvest should be more profitable than that. So the organism you are culturing should be able to eat cheap food and grow. That's what basically that they're saying. If it's accidental release, so this is a bit of ecological thing. Natural water bodies, there should not be inverse environmental impact. Nothing you can write down. Should not be invasive. Should not be invasive. Should not be an invasive organism. Okay. Of course, then if you take these prawns are cultivated sometimes in the natural lagoon areas. If you go to a Negombo lagoon or Jaffna or wherever, you are going to see these are isokotu keladiyo. So they are of course very close to the natural habitat. Okay. So if they most of the time they escape. So when that happens, they should not be able to, they should not damage whatever the organisms that are around them. Okay. should be able to tolerate high population density and grow well. That means they are able to live in crowded conditions. Can I, because what's our goal? To have a large number of organisms so that we take a lot of food. We can take, I mean, the harvest is high. So if the organism stops reproducing when the tank is sort of filled with organisms, that's not ideal. Ne? Because then you are going to need a lot of tanks. 30 tanks, that is not economical. So it should be able to grow in crowded conditions. Okay. Having resistance to common diseases. So immunity it should satisfy consumers by taste, nutritive value, texture of flesh and appearance, body color or color patterns. Right? So you know there are certain exotic fish, the tuna species, there are, I mean, there are so many certain specialized dishes that you make, food items that you make using certain fish, Japanese tuna fish, right? So those are the things that should be, those are the qualities of a good culturable species. Okay, so you can write down once in, you can highlight or small phrase that they should withstand the climate. They should grow well in prevailing physical and chemical parameters. Should be easy to breed. Uh, techniques of incubation should be techniques of should be available. Okay, then they should be hardy. They are resistant. Okay, the food and feeding habits are known. They should not reproduce in grow out tanks. It is ideal that they reproduce late, sexual maturation late. They should accept formulated food. Should be an efficient conservator of economical food items into, I mean, they should be able to eat economical food. They should not be an invasive species. They should tolerate high population density. They should have higher resistance to common diseases. Okay, then they should have these sort of consumer factors. They should have a good taste. Okay, food items. So those are the fish or the aquatic organisms that we eat. Right? We have some other aquatic organisms. Could be mainly fish, but it could be some starfishes. Some species people have sort of lobsters. Different people have different aquariums just for the beauty of it. So that's what we are going to talk about next. Any questions? Online people, any questions? Ornamental fish, okay, having century of years, okay. Initially, the fish that had some colors were collected from the wild and maintained in garden ponds, okay. Right. What are the reasons? You can highlight. What are the reasons that people have ornamental fish? Why do they keep them? Um, attractive coloration. Oh no, wait, hold on. People keep fish in their homes, such as for decoration, children's education, enjoyment, relaxation of elderly, health of affected individuals, prosperity, fortune of home of humans, and for the collection of rare species, and also to propagate them. Right. These are some reasons. Okay. Of course, you can have some fish tank for your decoration in your living area. Children can have certain pets. You can keep fish as pets. Okay. Then there has been some evidence that people get a distraction by watching certain fish in the tank. In Hammer Hospital, if you go to any private hospital, you can see in the waiting area there's a fish tank. No, that is because people can just distract. You can just stare at it. No? Stare at the fish. Easy to breed rather than writing the whole sentence. Should be easy to breed. Which instead of which sentence? This is also a good keyword to write. So that sufficient number of eggs or fry could be obtained. Okay. It can be a keyword as well. Sufficient amount of eggs or fry could be a keyword as well there. Okay. Okay. Right. Okay. No need to remember scientific names. නිකන් ඔය දන්නවනේ ගප්පි ඉන්නවා, black molly, platyl, discusලා, fightersලා, gurami, you don't these things, goldfish. Okay, koi carp. I just keep a few things, few species in mind just to be safe. What is the fortune of foam? Have you heard? There are these Chinese, what the arowana or someone, නිකන් golden කෙනෙක් ඉන්නවනේ, the golden arowana. There's a fish that is sort of golden colored and costs about a few lakhs, one fish or a couple of fishes. So it's believed that if you have a tank full of them, it's going to make you successful. Okay, golden arowana, I think they are kind of gold color and tadi. It's a gurami species, I think, right? They are a type of gurami, but some certain beliefs are there. Okay, and it's a sort of a bit of a sign of your wealth also, if you can buy and maintain them. Okay, so we come to aquarium. Aquarium, there's a definition. There's a definition of an aquarium. Can you highlight, Metin? Container that is designed to hold water in which living organisms can be maintained over an extended period of time. Right? So, if you have just a jam jar full of simple bottle containing some water, yeah, aquaria can vary tremendously. But empty jam bottle that is used to keep a male Siamese fighting fish to multi-million liters commercial exhibit tanks, right? So, but ideally, aquarium is a place where you can keep fish for a long time, within extended period of time, so that the conditions needed for the fish are provided. Okay, so you can design to hold water. Living organisms can be maintained over an extended period. That's a keyword. Okay. Okay, then maintenance of a home aquarium. So if you jump back to the tute, I have given you a diagram of a small aquarium, freshwater aquarium. What are the things? If you haven't had a fish tank in your home, then you might not know, but of course everyone would know this, right? To provide aeration, you need to have an air pump. Okay, because it's a small container, no? So the fish are going to run out of oxygen at a certain point. So you have to keep the air circulating, right? Then, these are not all needed. Okay, depending on the fish you have, if you are having tropical fish, right, you might need to maintain the temperature at a certain temperature. Okay, so you might have to have a heater and also a thermometer to monitor the temperature. Then under gravel filter means through the gravel you can filter the water, as well as you can sort of suck out the fungi and also the fish like a feces settle. So that is sort of sucked underneath by gravel filters. Then you can have a light source. You need to have a cover to cover the thing so that debris doesn't fall onto it. And there's a water filter as well to keep the water constantly filtered. That's it. I take a rough idea. We'll take down some notes here. Okay. So come back to the resource. Activities that should be carried out daily, weekly, fortnightly, and monthly. Fortnightly means 2 weeks, every 2 weeks. These are general knowledge. Okay, so we'll quickly go through in order to maintain environmental conditions. Okay. Hi. What are the things you have to daily do? I don't know why they are in the syllabus, but they can be questioned. Certain questions, MCQs have also been asked. You have to feed the fish, of course, right? With a balanced diet, following a correct feeding regime. So depending on the fish species, it could be 2 or 3 meals. Okay. Check on the status of the health while feeding. Remove affected individuals. Right? If you feel like a fish is having a disease, you shouldn't keep it with the other fish. So you have to remove it. Okay. So you have to keep him quarantine another tank or a basin. Make a bit interesting concept. Allow the fish to adapt for changing intensity of light. Fish are very sensitive to light changes because why? They usually live underwater. So the light changes very slowly. The sun sets and the sun rises very slowly. So they don't sort of they don't go inside or something. So they live in water. So they are only adapted to the speed of the sun. Okay. So we shouldn't switch on or off lights like as we want. Okay, we should allow them some time. This is valid for certain fish species. Okay, certain saltwater fish and saltwater unknown fishes line. That is sort of marine fish. Okay, so switch on the light of the aquarium several minutes after the room lights have been on. Okay, so if you have an indoor aquarium, okay, now it's getting dark. You switch on the lights in the room, but you don't immediately switch on the lights in the tank, because the light of the room is just enough for now. Then once the fish are adapted to the light in the room, then you switch on the light of the aquarium. Okay, then switch off after the room lights are switched off. Then in the morning you switch off the lights, then you switch off the aquarium lights, because the fish has to get adapted to the darkness again. Okay, before natural light sets. Make a kipata kolvithiyagana that you shouldn't immediately switch on your aquarium lights. You have to follow them after the room lights. Okay, then what are things you have to do weekly? What are things you have to do weekly? Should not be fed 1 day per week. Okay, right? But there are some fish. Okay, some fry and fingerlings. You give them a big meal once a week in very rare species. Okay, some species, so that they sort of they have enough time to sort of digest that. Okay, then every 2 weeks, what are things we have to do? Every 2 weeks you have to do a cleaning regimen. Right? Sati Degasaryak, you have to do a cleaning regimen. You have to clean fortnightly as well as monthly. This is sort of like a half clean. Fortnightly. Every 2 weeks you have to do like a half clean. Monthly you have to do like a full service of the tank. Right? The full clean. Okay. So fortnightly you switch off the aeration. That means the air pump you switch off. Okay. Then what do you do? Under gravel filter medium, you sort of rake or stir up. Again, if you don't have so in the gravel, there could be between the gravel, there could be debris, fish like feces and excess food. So what you do is you sort of you stir it up. Right? Then what's going to happen? The debris is going to come into the water. Okay. Then what are you going to do? You scrape off excessive algal growth from any ornaments that you have, and also from the glass. Okay. Then you allow the debris to settle. Then while you are siphoning off that water, you can take out that sort of dirt. Okay. You remove 20 to 25% of aquarium water, not more than that. Okay. Then you replace the volume with fresh water where you match the temperature, pH, and hardness. Okay, so this is very important. Have tap water you don't directly put. What we usually used to do in the home aquariums, we take tap water and we keep it for some time in the room, right? So that the excess chlorine can sort of dissipate, right? Have you heard? Right? If you had an aquarium, you might have it, right? So you take tap water and you will keep it outside on a basin or something for maybe about 2-3 days, so that the excess chlorine can sort of dissipate. Okay, but these professional aquariums, they sort of even measure the pH of the water and try to match that. Okay, switch on the aeration. Okay, that is done every 2 weeks, right? So you only remove 20 to 25% of the water. It's just a half cleaning process. But then monthly, you are going to completely remove all the water and you are going to clean this. Okay, you are going to take some water from the aquarium into another tank. Then you are going to take out all the fish. Using a hand net. Then, very importantly, you are going to put them in a smaller container. Yes, you have to have aeration. Or clean you need to give that second home some aeration. So do we have to remember these steps? Which steps? The aquarium maintenance, yes, aquarium maintenance can be questioned. Okay, rinse the rooting medium, right? You take out the root gravel and you can wash it. Okay, remove the aeration, scrape off any deposit. So around the aeration there can be algae formation. Then air diffuser stones, those are, I told you, under the flow. Okay, and air diffuser, sorry, the air diffuser stones, have you seen? I don't have a photo of that. Sometimes from the air filter, the air comes out through this sort of a, they sort of resemble stone, so that the beauty eකට මේකකින් තමයි you have connected the air, sort of the tube from coming from the air pump, right? So the bubbles come out of this sort of a, so the end of the tube is connected to a sort of something looks like a stone, right? So that is the air diffuser stones. So it's good for the beauty, and also it looks like, I mean, so that is, from out of that you get the air bubbles. Okay, so that the air bubbles get dissolved in water and you get oxygen for the fish. Okay, introduce the siphoning tube under the filter plates and suck out the accumulated organic debris. So under the filters, so these are sort of professional equipment that they are talking about. Haridu? Siphoning tube, that's like a vacuum cleaner for the water. Okay, to clean under the these filter plates. Okay, then you check the light source, okay, and drying leaves from the aquatic plants, because they are going to decompose and it's not good for the fish. Then thin out and dry the plants, okay. Rearrange the filter plates and filter the medium and the filter medium. You again put the aeration and you fill the aquarium halfway. This is important with within aged aerated tap water. That is called aging, aging the water. That is to mainly to remove the chlorine from the water. Reintroduce the fish with the water and then fill the aquarium to the original level using aged clean fresh water. Okay, understood? This is how you maintain an aquarium. Okay, so I think there was a question. So can you understand? So these are the under gravel. So you can see this is the gravel layer. So there's an under gravel filter separately in this one. Okay. So I think there was some questions. So you can see we are going to cover dengue, stem cells, other things. Okay. Dengue genome project. Amiti Mekanaban Tagala carried out monthly when maintaining aquarium. This is an ABCD type question. Okay, tell me the answers. So you, ask me. You have to remember these steps in order to answer these kind of MCQs. Okay. Okay. Right. Easily. What is the answer? Send me the answer. Yeah. ABD. Okay. Okay. Any other answers? Okay, A. Cleaning of air diffuse stones. That is monthly, no? Yeah. Monkey? Air diffuse stones, we need to know. Okay. Pruning aquarium, that is also once a month. Ned. Algae grown on glass surface, that is, you can do it every 2 weeks. Yes. Not done. Organic debris under filter plates. So, filter plates only after 1 month. Yes. Removing diseased fish, you can do that daily. You should do that daily. Yeah. That is, if you wait monthly, okay? Understood? Yes. Simple enough. Uh, yeah. Right. So this is in this paragraph. This paragraph is nice. Okay. So pay attention. Uh, no matter how much time. Okay. Excessive algal growth, poor water quality. So they are in this paragraph. They have given some problems and what is the problem. What is the reason for the problem and also how to solve that. Okay. So pay attention and highlight. Okay. Water turning green. So green problem megakna. Water is green, green algae, and on the side glasses, that is due to too much light. Can you highlight the problem and the reason? Green water is becoming green, and green algae is due to too much light. What do? Too much light, na? You reduce the light received, okay? Intensity or the duration of light followed by a partial water exchange. Okay, then if you have you seen in the these outdoor fish tanks, a green color nil. If you have had an outdoor fish tank, glass one in the outside, it is turning green very commonly because of the excessive sunlight. So that's one problem. Then brown algae issue. If you are facing a brown algae problem as encrustations, okay, and in the aquarium deco and the side glasses, it's insufficient. Green, okay. So there is your insufficient illumination. So what do you have to do? You have to arrange a better light source. Then the third problem, okay, you can have blue-green algal mats. Blue-green algal mats. Now, this mats is a key, nice word, okay. Indicate a high level of organic pollution. That means either too much food has been given, or you haven't cleaned it, so that the fish waste matter is accumulated on the aquarium. So you have to physically remove them by during cleaning, and also you have to change the water, and you have to review the maintenance routine. So sometimes some fish tanks, if you have a large number of fish, you have to maybe do the things you do monthly to weekly. Sometimes special situation. Sorry. Okay. Whether too much food is provided, inadequate filtration or aeration, overstocking. Overstocking can you have more. I mean, higher number of fish than you should. Okay. Very good malu dala. So you better take a bigger tank or have 2 tanks. Okay. That's it. Then these diseases. Okay. Few diseases. I mean, they have never been questioned, and it's a bit unlikely, but we'll just go through them. Okay. Let's see, Manadakian. Invasion of fish tissues by a disease causing agent, it could be virus, bacteria, fungus. It's very like us. Okay. These are fish, no? Multiplication of it in the fish tissues and increasing population may lead to the development of an infectious disease. Okay, could be avoided through better management practices such as BMPs, better management practices. Can you highlight that? Okay, maintenance of water quality, correct stocking density of compatible fish. Right, you have to do a good research before you stock. Some fish species are not meant to be together. Either compatible plants. Samahara plants can produce certain sort of products that can be toxic to the fish. Okay, so you have to do a good research, correct feeding regime, and also correct biosecurity measurements. Make a highlight. Can what are the biosecurity? Okay, if you bought some new fish, you don't immediately put them into your big tank. Okay, what do you do? Quarantine. Highlight that. Quarantine the new fish, plants, and other aquarium deco. Okay, if you bought some new fish, you have to keep them on a small aquarium. Could be another bucket with some aeration for a few days to see if they are developing some diseases. Okay, some symptoms. If that happens, you have to not add them to the main aquarium. Okay. Common diseases of freshwater ornamental fish. Okay, bacterial fish rot. I think, of course, right, gill rot. Have you seen this fish? That is bacteria. Sepsis. Septicemia means that means you have bacteria in the fish blood. Okay. Columnaris, I don't exactly know what is caused by that. Mycosis. Mycosis, of course, it is fungi in nature, right? Mycorrhizae, mycos. Myco is fungus, no? White spots disease, these things you don't need to remember, okay? Parasites, certain parasites, okay? Trichodinosis, okay? I'll check and tell you what sort of parasite this is, okay? Gill infestations could be, flukes. Flukes are type of worms, okay? These flukes can infect us also, that are found in usually freshwater bodies, okay? They are types of usually platyhelminths. Certain fish can get infected by, this is a worm disease, okay? Worm disease, okay? Right? Those kind of things. So, when you have time, you can shift to the tute. No need to exactly, but I would prefer if you make a small short note of the problems, okay, of the problems, because it's given nicely in your resource, the things you have to do daily, fortnightly, monthly, but I would prefer if you make a small note here. What is the cause for green, water turning green? Okay, what is the problem? Too much light. Okay, podi note okay, water turning green is too much light. Then if it's water turning brown, that is due to inadequate light, right? What else was there? Blue green, blue green seen at the, yes. If the water is turning sort of blue green, that is organic pollution. Organic pollution is high. So I want you to add the correction mechanism as well, what you can do to sort of correct it as well. Okay, make a small note there. It will be easier for you to memorize. Okay, could be asked in an MCQ. Blue green algal mats, brown algae. Okay, the important things are actually the things you have to do. So green partial water exchanges, okay, those things, and review the feeding regimen, adequate stocking density, those things you have to have a small idea. Okay, understood? Do that in your own time, right? Tonight itself, do this. Okay, other atom applied to a bigger part, you can finish a bigger chunk of applied biology. So this is kind of something we learn in environmental as well. Mainly, we are talking about the invasive nature of certain ornamental organisms. Okay, fish. So, what are the benefits? Earlier, also we talked a little bit. Species conservation. So some people have sometimes in their personal homes very rare fish. That is good. Okay, even could be illegal, but depending on the context, some fish species that have been protected from becoming extinct due to this ornamental value of them. Okay, and also production of species that are difficult to obtain from the wild. Approximately 90% of freshwater ornamental fish traded globally are done in captive conditions. There is some environmental benefit or derivation of environmental damage via those breeding programs. Okay, because we breed them under captivity, that means you are not catching natural organisms. You don't sell them because 90% of the freshwater fish you breed them and then you sell them. So that's a good thing. Okay, because you are not disturbing natural ecosystems. Understood? Ameti Nami, golden arowana, and tiger barb. I have seen golden arowana and haven't seen what's a tiger barb. Okay, that have been conserved via ornamental fish protection. Yes, they were under the threat of extinction. And also, in addition to cellular hobbies, fish are also being reintroduced to habitats from which they have been eliminated. Okay, because of the worldwide trade of these ornamental fish, sometimes they can be released back into their normal habitats. Understood? Then these are the negative things. Listen, okay, these are the negative things. Could affect a wide range of native organisms from zooplankton to mammals. Like a chutim organism to the biggest mammals in the ecosystem, they can affect due to the invasive nature. This is a keyword, okay? Invasive nature. With imported fish, non-engineered disease-causing agents may also come into a country, right? That is, you can import together with when you are importing a certain ornamental fish. If that fish was infected, you are getting a nice new bacteria into your country as well. A bacteria or a fungus could be a parasite. Then what a lot of people, so today when I was making this tube, when I search for certain fish diseases, only menne fish disease amoxicillin කියලා තමයි Google එකේ, right? That's because all these could be not very high educated people, people who have aquariums. What they do is whenever the fish has a disease, they put amoxicillin into the fish tank, so that they think, okay, it is going to be the solution for all fish diseases because it works maybe 5 out of 10 times. Okay, they are widely using it. So broad spectrum, so amoxicillin is a broad spectrum antibiotic. No, it works against a large number of bacteria as preventative or therapeutic. Sometimes very hondra kila they use before the fish gets a disease, they add antibiotics so to protect them better. Okay. And treated water containing antibiotics or other chemicals could be released to the natural environment. Okay, that may cause antibiotic resistance. Can you highlight this? I have to, I mean, antibiotic resistance is a big problem everywhere. Could be now then. Because when we are treating humans with antibiotics, the resistance also builds up. But may they malu hadanna gilla antibiotic resistance build that's such a wasteful thing. Right? Because amoxicillin is a very, very useful antibiotic and it is also one of the most what is say as abused antibiotics. Okay. Very soon it's going to be completely useless because of the resistance. Okay. So that's it about aquaculture and ornamental fish. Okay. Any questions? So we have some dengue, filaria, etc. will come later. Metin, make kanban taggala. Question number 5. May contribute to keep the immunity of ornamental fish at a high level. Immunity. What can you do to maintain their immunity at a high level? What happens when the antibiotic resistance is high? So bacteria will not answer to the antibiotic. So you won't be able to kill the bacteria and the bacteria will continue to grow. And then what's going to happen? You are going to have unhealing bacterial infection. So you can have a bacterial infection that is not healing. So it can go into your blood, it can infect your organs, and it can lead up to sepsis. Sepsis in humans, it causes low pressure, cardiovascular sort of, it's going to cause hypotension, and you are going to die. Okay, so antibiotic resistance. Unless you discover new and new antibiotics, that's one of the biggest risks. Yeah, what's the answer? Okay. May contribute to keep the immunity at a high level. Okay. Correct feeding regime as a biosecurity measure. Is correct feeding a biosecurity measure? What do you think? Where was this? Okay. Okay. What's up? Oh, yeah. Okay. What are things prevent contamination? You can quarantine, no? Biosecurity measures also you what comes under is quarantining. So correct feeding is not a biosecurity measure, no? Right? So you can't take this. Entire volume of water fortnightly, you don't do that. Ne? How much do you replace with an Israel? You can write down. How much water percentage of the water do you replace? 20 to 25%, no? The turning of aeration at night. Okay. Malu rattles magannad. Okay. Right. They breathe at night as well. Keeping the lights of the aquarium switched on continuously. No, your fish should have a daily rhythm as well. So you should try to mimic the normal light conditions. False. Maintain the correct stocking density, yes, of compatible fish or plants and fish. When the fish are overcrowded, the diseases are going to get spreaded easily. Correct feeding mechanism can prevent some diseases, but not infectious diseases. I don't know if fish get diabetes or something. Okay. Um, okay. Water quality, correct stocking density, correct feeding mechanism can come under. Oh yeah, feeding can come under immunity because yes, because the good nourishment. Yes, good nourishment can improve the immunity, but it is not a biosecurity measure. Can you understand? It's not a biosecurity measure. Biosecurity means you are sort of the quarantining is the biosecurity measure that we have to consider here. But obviously, answer number 5 is the obvious correct answer. No? Yes. Hi. Okay. Any questions? All right. We'll quickly move on to then our. These are all external age. අපලගේ resource book එකේ PDF එක මේක delete කරත් කමක් නැහැ. Right? All these you don't need. Useless. Okay, no need. Within flukes, if you just want to read them, this you can talk about these flukes. Okay, right. We're done now. Right, we move on to a bit of botany. Okay, a bit of plant propagation. These kind of things could be a bit boring, but okay, questions are there. Okay. Nursery management. Nursery management can you are not your nursery school, but for the plants. Okay, so the nursery for humans and plants mean the same thing. You are growing small organisms. Growing means what? Yeah, developing. Okay. There's a definition. Highlight. There's a definition. It's a specialized place for young plants and seedlings to be produced for transplanting in another place. So it's like a training camp or it's a school, right? Where the plants grow up to become independent adults. Okay. Plant propagation and nursery management can be done in the field, orchard, forest, or in a protected environment. Yes. So you can have a nursery in all these places. Okay. But there are specialized nurseries that can be constructed in greenhouses, polytunnels, right? A polytunnel is just to main sort of protect it from direct rain and direct sunlight. But a greenhouse is a much more different thing. We'll talk about what's a greenhouse is. Or you can have a complete lab setting, right? Multi-million dollar companies can have these sort of labs dedicated to propagating their plants. Okay. The main requirement of a nursery is providing optimum favorable conditions for the germination and seedling growth. Okay. Which is going to provide healthy, vigorous, evenly grown plants. Okay. So make it better to keep that in memory. All right. What is the main purpose? Is to optimum of favorable conditions for the germination and seedling growth. That's what you are going to do with a nursery maintenance. Okay. What are the issues? This is good. This paragraph is good. Okay. This paragraph is about issues. Issues faced by faced when you are trying to maintain a nursery, right? Uh, Issues within management of light. Okay, you should be able to. I mean, the weather is not going to be working according to your timetable. Okay, so you should be able to. So the light conditions changing is a big problem. Then soil management, soil type, properties. Okay, and maintenance. Some a lot of you have a nursery, and once after a big rain, the nursery soil has been completely washed away, right? So you have to think about those things. Then this is the biggest thing: pests and diseases. Okay, protecting from them is very difficult. Okay, water management. Okay, you have to conserve water if you are living in an area without adequate water. Nursery structure and environmental controls. Okay, so nursery structures. Hurricane එකක් හරි strong wind එකක් වගේ આવොත් the nursery could be blown away, fallen down. Okay. Environmental controls, protection, cultivation, protected cultivation versus open air. So greenhouse and these polytunnels are kind of protected cultivation, or you can have just in the open. Okay, if it's a good season. Okay, then soil management is a must to get a quality product. Soil texture, structure, and organic matter content influence the soil condition. All right, make up put a highlight. What are the parameters in soil? Soil texture, structure, and organic matter content. So if you take a sandy soil, you are not going to have much organic matter, right? If you take a clayey soil, then the water retention is too high, no aeration, air available for the roots. Pest. You can have biological, cultural, physical, or chemical things to get rid of pests, right? You can have biological controls, you can have natural predators. Okay, or a birds lover, you can attract birds into your sort of cultivation area so that they eat the insects. Biological you can have chemical pesticides. Okay, physical things that make some noises, even I don't know, right? Okay, to get rid of the pests. Okay, water management and humidity control. Okay. The type of irrigation used depends on natural conditions of the area, soil type, slope of the land. This is important. If you are cultivating in Norelia, you have to think about the drainage. Most of the water you are going to provide is going to just go away. Okay, that's therefore. Have you seen in Norelia where they have this sort of a leeks or carrot? They are going to have this permanent water pipes, tubes that are going through all these what do you call them? Blocks of carrot and leeks, so that they are having a trickling of water all the time. අපි වගේ බකට් එකකින් වතුර ගැහුවා කියලා ඉවරලා they are not going to remain because it's a hilly area, no? Right? So you have to think about those. Then water availability. If you are cultivating in a dry area such as Jaffna, you have to conserve your water. Okay. Nutrients are supplied to the seedlings, although there's a number of ways to apply fertilizer. The type chosen depends on the fertilizer available. Then now you know some ways. I mean, from microbiology, you know ammonia should also be always be hydrated. No, why is that? Why should ammonia be always be hydrated? If you apply ammonia to the soil, it's not going to remain as ammonia for a long time. It should become ammonium very fast, otherwise it's going to dissipate, right? That's why ammonia apply kala you have to water the whatever the cultivation. Okay, then other issues. So lack of access to modern technology because they are farmers. Okay, lack of financial incentives to further improve the nursery. Okay, then cultivation facilities, storage, shortage of planting and propagating materials. You can't obtain adequate seeds. So again, there could be problems. Inadequate knowledge. All those are issues, other issues. So Methana, there's 4 issues. Then here there are 1, okay, 1, 2, 3, 4, 5, 6. Altogether 10 issues are there. So try to remember at least 5 or 6 words, okay, to write down in the structured essay paper. Okay, at least remember few of the issues. Understood? Any questions? No? Going too fast? No? Not going too fast? Okay, nothing much. Uh, then methods and rationale of protected cultivation of crops. Right, protected agriculture. So we are going to move on to talk a bit about these polytunnel and greenhouse cultivation, those sort of things. Okay, let's see. Various types of plant growing structures they could provide a more favorable environment than the open air. Okay, because you can control the conditions inside this covered. You can control the temperature, humidity, right? Simple terms, Metin, there's a definition. Haridha, highlight this. What is a protected cultivation? Growing crops under controlled environmental conditions. Okay, that's the definition. Okay. Protected cultivation technology used to protect plants from adverse climatic conditions, could be wind, heavy rain, mist, by providing optimum conditions to achieve maximum yield and the best quality. Could be done in a greenhouse, okay, highlight, okay. Depending on the covering materials, different structures can be used, such as polytunnels, okay. Protected cultivation is mainly utilized to grow perishable horticultural produce such as fruits, vegetables, and ornamental plant. They are usually expensive, within bell peppers, strawberries, right? Those kind of sometimes imported seeds, and sometimes they are genetically modified. Okay, those things are usually grown in protected cultivate in the form of protected cultivations. So did you highlight the definition of protected cultivation? Yeah, clear? Okay, so protected cultivation, right? I didn't have to sort of, yeah. A simple form of a greenhouse is a structure where it is covered with a material that allows light to penetrate inside and reach the plants. Okay, so you can see a few examples. Okay, it's a structure where it is covered with a material that allows light to penetrate inside and reach the plants. Okay. Okay. Please remember some examples of things that are grown routinely in a greenhouse or a polytunnel, bell pepper, tomato, salad cucumber, cauliflower, lettuce. All these are sort of a visual sort of crops, right? අපි ලස්සනට තියෙනවා, right? In the salad or in your burger, the lettuce it doesn't have a taste, right? මොකුත් රහක් නැහැ. It is just to sort of visual appeal එකට, right? Then salad cucumbers, those sort of very fat ones that could be grown in these sort of polytunnels. Strawberries, of course, they should be all protected. Ornamental plants such as carnations and orchids, yes, they have a specific moisture, they have a specific humidity, specific sunlight conditions. So those you can control only in a greenhouse. Okay. All right. Then we move on to important concept. Okay. I have drawn some things in my own PDF. Okay. Anyway. All right. So come to this is a very important topic, tissue culture. Either pay close attention. Principles and its importance. Okay, we'll read about this and we'll go into the tute and make a small note. Okay. Two of the most important areas under biotechnology. Okay. This is okay. Tissue culture definition is this one. Okay, is the ability to establish plant tissues, cells, callus, and protoplast, and plant organs, embryo, shoots, and roots in an aseptic in vitro culture. Can you highlight this? That's a definition. Okay, so this could be questioned. Okay, so I think so. Plant tissues can have several developmental stages. Okay, so you originally can have a protoplast. You know what's a protoplast? No? What do you mean by protoplast? It's a plant cell. Okay, what do you mean by a protoplast? It's a plant cell without the cell wall. Okay, a protoplast you can write down here with a pencil. Okay, you don't need to know that. It's a plant cell without the cell wall. Okay, then you can have the cells itself, and also callus is like a ball of cells that is like a first step of the tissue culture, right? Once you culture a certain cell, it can develop into a callus and then it will grow into certain proper tissue organs. What are the proper organs? You can have embryonic stage. From that embryo, you can have shoots and roots. Okay. Then what do you mean by this aseptic? Aseptic means, of course, it's a culture, right? It's a culture. That means you have to provide nutrients to it. No? So when you provide nutrients, there is going to be the risk of other things growing in this. When a bacteria, fungi, all the other things could grow in this same culture. So you have to keep them very protected, usually in lab conditions. So this is an important word, aseptic. In vitro means what? You do it outside cells, okay? Outside. The most common benefit, also important, okay? What is the most common benefit? You can clone or mass produce genetically identical organisms. So imagine if you have produced a genetically modified organism, you made a very good genetically modified organism by complicated gene techniques. So you have to keep on obtaining new plants from that organism. So in order to do that, what is the best way to do that? So you get exact copies. You do tissue culture. Okay. So I think we jump to the tute. Yeah. So tissue culture maker, you can use this as a reference, small reference. Okay. So you can take usually any parenchyma cell you can take. Okay. Then what you do is, this is what I called as callus formation. That is like small floating balls, small groups of floating plant cells. Okay, when you place it on a nutrient medium. Okay, so this medium can contain growth factors such as auxins and other things. Okay, so it's an unorganized mass of cells. It's a callus. Okay, and now callus is a, you can draw an arrow. Okay, it's an unorganized mass of dividing plant cells. Okay, right. So you can see, then from the callus, there's going to be organ production and organ formation. So you develop shoots, then you produce roots. Once you get roots, what can you do? You can take it out of the whatever the culture medium. You can put it into a protected cultivation, such as a greenhouse. Then you can have another plant. Exactly same. This is because this is why this is asexual. No, this is completely asexual. No? Yeah. You are utilizing what property? You are utilizing what property of the tissues, plant tissues. They are inherent. If you take a parenchyma, what do you learn about parenchyma? They retain their, yeah, they retain meristematic ability. They can keep, they can divide if you give the right conditions. Okay. So we'll finish talking about tissue culture. Right, Metin, the main concept behind plant tissue is totipotent. Can you highlight this? What is the definition of totipotency? Megathyroid kargan. Definition of totipotency. A single cell has a genetic program to grow into an entire new plant. You can get an entire organism from one single cell. So, of course, these people you know, Schleiden, Schwann, and Rudolf Virchow, they introduce a cell theory. Then there's another definition, megathyroid kargan. This is the definition of a tissue culture medium for plants. So we had another culture medium for microorganisms. Okay. Inorganic salts, organic substances, water, and gelling agent. Then organic substances must always include what? A carbon energy source, usually sucrose. We usually provide sucrose because most of the plant physiology is based around sucrose. You know, phloem transport when it sucrose, no? So sucrose is very ideal in for most of the plants. Okay, sucrose is ideal. Then what else? Plant growth regulators such as auxins and cytokinins, vitamins, miscellaneous compounds. You need to have agar because agar provides a physical support for the explant. Okay, increase the aeration. Explant is the first cell that you obtain, right? You need to keep it on the surface. Okay, it cannot sort of get the air. So you have to have a physical support through the agar. Also increase the aeration. Murashige and Scooge or MS Medium killer, there's some commercially sort of they have given their names and you can use them for commercially to cultivate. Okay. So jump to the tube and we can write down here. Okay. Another plant tissue culture medium plant tissue culture medium. What do you have in plant tissue culture medium? Yes. What do you have in plant tissue culture medium? So you can say organic substances and inorganic killer you can divide into two groups. Okay. So under organic number one, you are going to have carbon energy source. Okay, energy source, which is usually sucrose. Okay. Then what else? Under organic, what else do you have? Then I'm just going to say plant growth regulators. such as one other auxin, cytokinin 3, vitamins. Okay, then you can have AGA. AGA is an organic thing, no? Then inorganic, what do you have? Macro and micronutrients. So you know macronutrients, okay, and micronutrients, they are the inorganic compounds. Okay. So take down this list, and under AGA, I want you to write down what are the functions of AGA. What is the function of AGA? Physical support and aeration. Okay. The AGA provides physical support for the explant and also increase aeration of the medium. Okay. Then also write down here an example for some commercially tissue available tissue culture. Could be sometimes questioned in the structural essay. Some names Murashige and Scooge and MS medium. Okay, those things pretty idea okay. Okay. Haridhi, come back to the resource. This list could be questioned. Huh? What are things found in a plant culture medium? Okay, right. Importance of tissue culture technique. Okay, some points are there. We'll highlight as we as needed. You can use them for rapid multiplication because it relies on mitosis. Okay, you can obtain plants rapidly because you don't have to wait until another that plant reaches reproduction maturity. Ne? You don't have to wait until it reaches produces sperms or ovules. You don't need, because you can take a tissue and you do the tissue culture. You can obtain a large number. Mass propagation of specific clone. So if you produced a large number of clones, then from that clones you can screen and take out the organisms having the best qualities. Then you only do tissue culture next time for the best ones only. Okay. Genetic uniformity, I told you, because it's an asexual one, you don't have to worry about mixing other genes. Okay. Then you can do genotype modifications. Okay, genotype modifications, because it's easier to tissue culture GMOs or genetically modified organisms rather than sexually reproducing. Then you can produce large number of plants in a small space. Lab, in a single lab, you can have several cultures happening at the same time. Yeah, pathogen free. Yes, you're doing it in a lab condition, right? So all the new plants that you produce are completely free of pathogens. So you can safely export them or safely give them to farmers. Yes, all year round production, because it's again inside a lab. You are not affected by what's happening outside. It's Vahina, the route, it doesn't matter. Okay. Amendment import. Produce plants which do not produce viable seeds. Some plants, they don't have a good seed production. So the best way to propagate them is by doing a tissue culture. Okay. So this paragraph is nice. Okay. Remember at least 4 or 5 out of the 8. Okay. Then example of tissue cultured plants are anthurium. Anthurium doesn't produce successfully seeds. Okay, so usually you right, I don't know who's culturing anthuriums. Okay, right, banana, pineapple, dragon fruit. Dragon fruit is very yes. Dragon fruit is usually imported as small seedlings. Okay, then you cultivate it. So after yeah, this tissue culture is like a sort of advanced asexual reproductive method, asexual propagation methods. There are other asexual propagation methods that you don't need a lab to do. Okay, lab behavior, you don't need culture media or other things. Those are grafting mainly. Then there are a few other things. Okay. So before that, we'll have to talk about the proper sexual propagation that is done by seeds. Okay, so if you can obtain seeds, that's good. That is the natural way to do it. No? Right? If you can propagate using seeds, that's the natural way. Okay, but it's not commonly done nowadays. Therefore, production of high quality seeds is a prime importance. Okay, what are the qualities of a seed? Higher percentage of germination and higher viability. Higher viability means they should be very sort of, I mean, they have a higher chance of becoming a good plant. Okay, free of diseases, of course. Okay, seeds should be free of diseases and should not be damaged. This is a definition that we encounter now, right? In plant form, we didn't have a very successful germination. Okay, it's the beginning of the next sexual generation of a particular plant. Can you highlight this? This is a definition. Seed germination. Then these three. What are the conditions to initiate? Very, very important. Three things. Seeds must be viable. Viable means they are alive when fertilized. Okay. Then you can write in pencil. Viability means alive and fertilized. Viable. Then it should experience suitable environmental conditions. Okay, the environmental conditions should be ideal for the germination to happen. Then existing dormancy should be overcome. What do you mean by dormancy? Certain factors that are preventing. Could be the factors? The seed coat can be too thick, right? Then the existing light conditions could be not ideal. Those kind of dormancies should be removed. Anthurium also can be okay. Seed propagation methods. Okay, is the preferred culture method. Okay, anthurium and orchid can be. I don't know if anthurium, but okay, you can highlight anthurium and orchid seeds are there. Orchid seeds are commercially available. Okay, very commonly done. Understood? Any questions? Vegetative propagation method. So that is all about seed or sexual based propagation. Small paragraph. Methoding, the most important things are this: how is germination going to happen? So better memorize this a bit. Okay. So that's all. That's there for you to be concerned about. Alright. We'll come to vegetative propagation. Oh, you are other asexual. So you know tissue culture is an asexual method. So that's a special one. What are the features of an asexual? The new plant is always genetically identical. Because why? You are relying on one set, what cell division? Mitosis. Ne? You are relying on mitosis. So because of that, it's always going to be completely genetically identical. Okay. So few techniques we need to know. Chutikala in I think in grade 5 also you had this for environmental studies or something, how to grow this sort of how to grow a new plant from a existing plant. Okay. And I think you had a subject from grade 6 to 9 or a PTS there was a subject that you cook things and maintain the school garden. You didn't have, yeah. Okay. That's maybe local syllabus only. Okay, number one, separation and division. Okay, right. Highlight the definition. Method where naturally detachable. This is a keyword. Okay, detachable structures such as rhizomes, bulbs, combs are used. Okay, so underground stems, underground storage organs, you can take them out that are natural. Again, in Sinhala, you call them moteyo again, right? That are sort of, so if you take a banana plant, around a banana plant, you can see small banana plants that are not actually small banana plants. Those are detachable small bananas that you can take them out and you can use them for to have another banana plant on a different place. Okay, so those are what we call as naturally detachable structures. So remember rhizomes, bulbs, and combs. Then the other part of this method is division. So separation, always keep in mind, you are not cutting anything. You are not cutting. This part comes out naturally. Galavila, you can separate in separation without cutting. But division you cut. Okay, keep that in mind. In division you cut. Cutting or dividing the plant into sections with both roots or with root and stems. Okay. So you cut things in division, you don't cut in separation. Keep that in mind. So alocasia, colocasia, wala comb you can take. Then runners, things that are running along the ground, spider plant. Stolons, right? Certain grass species. Okay. Can you highlight this? So at least remember one example from combs, one example from runners, one example from stolons. Okay, that's enough. Right, cutting. So we'll try to finish as much as possible. Okay, cuttings is the most important clonal regeneration. Okay, so adventitious root formation is a so mehmai. So if you take in, so one of the things you need to understand about division and cutting. Okay, in cutting you take, sorry, in division, I'm sorry, in division you take a piece of plant having both stems and roots. Okay, for example, if you take a runner, that means it has some leaves that is running, it has a stem that is on the ground, it has roots as well as leaves. So in division, remember, okay, it has both roots and stems already. Okay, you are just separating it from the mother plant. Separating means you are cutting it from the main plant and you are planting it somewhere else. Okay, but in cutting, Either it is the shoot you mainly take. Okay, so Metin, so adventitious root formation. That means whenever you have cut a piece of shoot, that shoot doesn't have no roots. No, then okay. So imagine it's like this. You take this piece of plant, and then when you plant it, it should be able to develop roots. Ne? Roots it should develop. That is called adventitious root formation. Whenever you missing the roots and you are planted in a soil, you should be able to regenerate root. Can you highlight that? You need to be able to produce adventitious roots if you want to do cutting. Meaning, then a piece of plant from the stem, leaf, root, or a leafy bud can generate into a fully developed plant. Then there's a definition. The roots arising from the stem, leaf, or bud tissue are known as adventitious roots. So when you plant a piece of stem, okay, or a leaf, that piece should be able to develop more roots and become a mature plant later. Okay, that's what you mean by cuttings. Roses, ficus, dracena, African violets, crotons. Okay, then all those stem or leaf bud cuttings or need only a new adventitious root system. For root and leaf cuttings, both shoot and root systems need to be developed. Can you understand? So imagine you took a shoot, right? The mevage shoot or a piece of leaf if you took, the part of the shoot system is already there, no? Then you have some leaves, right? But if you take a piece of the root, so it's just a piece of root, no? So it needs to develop both shoots and new roots. You should develop both shoots and new roots. Let's just keep that in mind. Okay, so leaf bud or stem cuttings need only a new root system, but you need for a root or a leaf cutting, you need to have both shoot and root systems. Understood? That is about cutting. Understood? Okay. Cut. Then layering. Layering you must have seen being done even in your home. If you, I mean, if someone in your home is interested in bit of gardening, okay, where stems are rooted while still attached to the parent plant. That means you haven't cut the piece completely separate. It is still attached to the mother plant, but it is going to develop roots. Okay. Some plants have a natural tendency to regenerate by self layering, forming adventitious roots on the stem where they touch the soil. Yes. If a piece of the stem bends down and touches the soil, from that place, some plants tend to develop roots. So, what we actually do is, so imagine you have a mehema, usually it is done for roses and hibiscus, right? In rose bushes, if you have this sort of a branch that is bending down, what you do is you bury that under the soil. You bury it under the soil. So there is going to be a soil layer. So this is buried under the soil. Okay, this is buried under the soil, this piece. Then what you do is you actually cut a part of the epidermis here. You cut a part of the epidermis so that the inside is sort of exposed to the soil. Then what's going to happen is roots are going to come out from that place. And then what you do is, since this part now has a separate root system, you can take this part out and you can plant it on the ground. That is called as layering. Okay. So while it is still attached to the mother plant, you make it have some new roots. Then you can cut it and plant it in a different location. Okay. Nitin, what is the benefit of layering? What is the benefit of layering? You can get a small number of new plants with relative certainty because the new plant is nourished by its parent plant until roots are formed. Then you can be certain that you can get a new plant because why? It it is unlikely to fail because it is still being nourished by the mother. No? Kardilavat, if it doesn't develop roots, that means that that piece is not going to die. Okay, so most likely you are going to get a new organism because it is still protected by the mother plant, means still connected to the mother plant. But you can't take many hundreds and hundreds of seedlings like in a tissue culture. Okay, it's only done to obtain a few new plants. You can do layering. Okay, then you have grafting and is that it? Yes, those are the propagation, plant propagation methods. There's only one more. Okay, grafting is joining definition highlight. Joining two separate plants so that they later function as one healthy plant having the best characteristics of the two parent plants. So it's you are sort of making a monster. You connect our Frankenstein monster. You take different organs from different organisms and you connect it. No? Different people like in hand, dekai, foot, dekai, again you connect, right? So in grafting also, you make a sort of a hybrid or a combined plant. Okay. So what are the two pieces you are going to need to make a new plant? You are going to need a root that is called as the stock. Then you are going to need a top part or a shoot that is going to be provided by that is known as a scion. Can you highlight? Root is known as the stock. The top part of the shoot is known as the scion. Highlight that. So you can say මෙහෙමනේ අපි කරන්නේ. So අපි මේක graft කරනවා කියලා. Imagine you have a tree that is rooted to the ground, a plant. Then what you do is you have a plant like this. You cut it from here, and what you do is you connect a shoot system from another plant. A shoot system from another plant. You do this because maybe this root system is very well developed. This root system is very well developed. So what you do is to that root system you connect a very well developed shoot system. You'd connect a very well developed shoot system. So you call this the scion. You call this the stock. Can you draw like using the pencil a small rough diagram? The stock and the scion, to remember that. Stock and the scion. Done? Any questions? Going too fast? No. I'm asking and I'm saying no. Ne? Too fast? No. It's not, I mean, වැඩි time waste කරන එකත් it's not ideal for applied biology. That doesn't mean you shouldn't study, right? Otherwise, it's going to be waste. But okay, we should keep it as efficient as possible because time is money these days. Not money for you, but it's running out. The time is running out. Okay. Floricultural practice found in Sri Lanka includes cut flowers and ornamental foliage plants. Cut flowers, that means the flowers you sell as bunches or bouquets. Okay. Then ornamental foliage plants means that those are the plants that you grow for the beauty in your garden. So they are still attached to the plant. Okay. Um, Anthurium and orchids and flowers, rose, dracaena, and begonia. Okay. Remember those things. Highlight. Okay, so there's a small error here, but I don't know if I haven't they corrected it. But rose is mainly a cut flower, but dracaena and begonia, dracaena and begonia, those are usually grown for the sort of the beautiful leaves. Okay, if you search them, you see dracaena and begonia, those are sort of they are grown for the sort of their nice arrangement of leaves. Okay, but anyway, we can't do anything. So remember anthurium and orchids as cut flowers. So you can sell them directly as flowers. And foliage, well, the Dracaena begonia. Okay. Any questions from plant propagation and tissue culture? Yes. Mainly, sort of just remember the important points without worrying as much. Okay. Hi. Make a question number 8. Question number 8 can ban tagga. Send me answers, online people. What's the answer? Yeah. A and B. Okay. In the floriculture industry, and how do you propagate them? Okay. Hibiscus, you can layer them. Ne? Yeah. Hibiscus, you can layer, right? You can grow underground and you wait for them to root and then you cut it and then you go and plant it somewhere else. Snake plant, using leaf cuttings, can you do it by leaf cuttings? Yeah. What do you think? Leaf cuttings, snake plant. So it's a cutting, no? So yes, very correct. Okay. Spider plant by grafting. Can you graft spider plant? Can you? Is it given? What do you do for snake plant is sorry, spider plant. Yeah, you use runners, no runners. So runners come under separation and division. So it's actually division. Okay, it's actually division. What do you do in division? You cut anyway. Okay, you do a cut, but the piece that you are cutting and taking out already has shoots and roots. They come with you. Okay, so that's false. Begonia, what do you do for begonia? It's again leaf cutting. It's cutting. So not separation. A and B am I correct? Yes. Yeah. Answer number one. Good. Okay. So better to remember the examples. Can you understand? Better remember the floriculture method and the example. Okay. That's a good way. Make a taggalaka. What are the conditions that a seed has to complete to initiate germination? That is from that section under germination taggala. Okay. What are the conditions that a seed has to fulfill? What is a checklist or the to-do list that has to be completed? Yes, for germination, the seed must be viable. It should be subjected to suitable environmental conditions. Then existing dormancy should be overcome. Existing dormancy should be overcome. Okay. The seed must be viable. The seed must be subjected to suitable environmental conditions, and existing dormancies should be overcome. Okay. So shall we continue direct straight until about 7:30 and finish? Yeah. I Let's see how much we can go ahead. Okay. Within 2 days, I want to finish this. Okay. We have already made some good progress. So we'll keep this going. Online people, any questions? Yeah. Yes. Okay, let's go ahead. Okay, we can move on to food. Okay, so we have moved from aquaculture, some bit of water organisms, to a bit of botany. Okay, gardening, and we are going to move on to the kitchen. Okay, we're going to move on to the kitchen. That's why we call this applied biology, right? You are applying certain biological concepts such as different types of reproduction, different certain. I mean, now we are going to talk about microbiological processes. Okay, so we are applying it in different scenarios. Okay, right. Most kind of foods are readily spoiled. So this is the last part of microbiology. We talked about food spoilage. Okay, so we talked about what's going to happen microbiologically about putrefaction, fermentation. Yes, yeah, those things we talked about. No, rancidity. Those things we talked about. So what's happening microbiologically, we know. What now we are going to see is how you can prevent that. Okay, how you can scientifically prevent that. Okay. Um, this is definition. Okay, hold on. Yes. Food preservation is the process of treating, handling, and food to stop or greatly slow down spoilage, the loss of quality, edibility, and nutritive value caused or accelerated by microorganisms. Nice full definition. Okay. Treating or handling food to stop or reduce. Stop or slow down. Okay, to stop or slow down spoilage. Okay, stop or why is it not? Okay. Stop or slow down spoilage, which is the loss of quality, edibility, and nutritive value. So, by just spoilage means you know this, right? The spoilage doesn't mean you it doesn't have a nice taste. Okay, spoilage can mean that you have lost the nutritive value as well. The food doesn't contain any nutrients. That means the food is spoiled. Okay, edibility means if you ate it, you'd be sick because of whatever the toxins or the alteration there. Okay, right. Right. Food could be preserved before undergoing spoilage using suitable techniques which could prevent unnecessary wastage and contribute greatly to meet the food requirement of a growing human population by eliminating avoidable losses. Avoidable losses are there, no? Right? Making more nutritive food items from low grade raw commodity using proper processing and fortification. Yes, you will see some examples where due to the process of preservation itself, the food has become more nutritious. Okay, because of the things you do to keep it for a long time, you already have made it more nutritious as well as sort of a side effect. Our main goal was to preserve the food, but at the same time, the food has become more nutritious as well. You'll see some examples. So, more nutritive food items. Then you can divert a proportion of. So, currently, food that is being fed to animals because humans are not able to consume, you can take it back to human consumption by processing it. Then you can fortify low grade food into nutritive animal feed. Again, so currently food that is being fed to animals you can modify so that you can feed it to humans. Then currently animal feed you can also improve their nourishment into nutritive animal feed. Okay, you can sometimes convert organic waste into animal feed. Okay, so everyone is benefited, animals, humans, everyone. Okay. Principles of food preservation. So what I'm going to do is, so whenever we are discussing a type of food preservation, I want you to tell me which principle is being used here. Okay, so earlier we did something like this, so microbiology. Whenever we are talking about an industry, what sort of microbial principle is used here? Is it the simply the microorganism itself? Is it a microbial process or a microbial metabolic process or a metabolic product? Right? Now, these are the principles of food preservation, right? These are very, very important 1, 2, and 3. Okay. What's the first one? You prevent entry. This is the key word. You prevent entry, then you prevent the growth and activity, then you remove or kill. Can you understand? Can you highlight the keywords? What is the first? You prevent microorganisms from coming at all. If they come somehow, you kill them or what? You reduce their activity. Sorry, you prevent their further growth or activity. That is the second thing. Third thing, you somehow kill all the existing microbes. So entry, prevent growth and activity, remove or kill. Three principles. So I want you to tell me in each food preservation method, what is the principle used here? Is it prevention of entry? Is it inhibition of activity? Or is it killing? Okay. Hi. Aim to prevent contamination in the first place and to remove or reduce the numbers. Okay. Uh, Achieved by application of physical, chemical, and or biological methods. Right. So some methods can be a mix of physical, chemical, or biological, or all three together. Right. Let's see. Microorganisms need a source of food and water. Then also they also need a suitable pH and temperature. So what we do is one or several of the living conditions needed for the growth of microorganisms can be removed. Okay, so you just make it less nice for the microorganisms. So then they will either be killed or their activity will be inhibited. Okay. Hi. First one, okay, very important and very old one. Okay, drying. Highlight one of the oldest methods. Okay, usually dry it in the sun reduces the water content of the food sufficiently to prevent and delay microbial growth. So there it is given. What are you doing by drying? You prevent or delay the microbial growth. Okay, so it is coming under the which principle? The second principle, reduce the activity by drying. Reduce activity because you are dry, water content is low. Okay, it is a second principle. Then you need to know the examples, grains. Most of the wet grains, including rice and all the other lentils, right? You buy them in the form of dried grains, no? Okay, that is going to reduce their, sorry, increase their shelf life. Shelf life means the amount of time you can keep them in storage. Okay, then what else? Wheat, rice, oats, rye, and barley are left to dry to prevent spoilage. Highlight those: wheat, corn, oats, rice, rye, and barley. Then some meat items you can do. Ham. You know ham. Usually you do smoking. A bit of smoke drying. Again, you hang the pork. Yeah, pork, right? Hams are made of pork. Yeah, pork. Is it beef? Pork, right? Yeah. You sort of smoke dry them by hanging them in specially sort of smoking rooms, but in traditionally they are just around the sort of the, what do you say, the chimney or the hearth. You just hang the hams and they are going to be preserved nicely over a long time. Okay, drying in ovens. Okay, so remember those grains and hams dry. Then another way, heating. So heating comes under, I mean, under heating you have several. There is pasteurization also comes under this, but we have already talked about pasteurization. Therefore, it's not mentioned here. So you know the exact temperatures of pasteurization as well. Okay. Then heating. Second one, majority of harmful microorganisms are killed at temperatures close to the boiling point of water. It's a preliminary step in many other forms because you actually boil the food before you subject it to other sort of preservation. Okay, it's a way of cleaning as well. You put it in hot water and you boil it. At the same time, you clean it. Okay, to heat the food to temperature sufficiently high to destroy pathogens. In many cases, food is actually cooked prior to their being packaged, right? So usually, if you are doing heating to the food, that means the food is going to be cooked, no? Right? It's inevitably going to be cooked. Okay. At pasteurization we have done. Then we go to the opposite of this. So, Hari, in heat treatment, what are the principles used? What are the treatment used? This is actually the third principle again, mostly. You kill, no? Okay. You kill. Okay. Freezing and chilling. The next one, freezing and chilling. So we can highlight a bit. Is perhaps the most common. Yes, freezing is very common. No, we do it every day in our homes, both commercially and domestically. Commercial freezing is done in different types of freezers and under extremely fast freezing or fast freezing. Okay, so there are two types of freezing that you need to know. Okay, there is extremely fast freezing and then there's fast freezing. Okay, so in extremely fast freezing, what happens is, you don't need to remember this, uh, between minus 18 to minus 30 degrees Celsius within 15 minutes you can bring, or in fast freezing you bring it down slowly in within about 2 hours. Okay, but anyway, if you are freezing something, it has to be in minus temperature, right? In commercial freezers, definitely the temperature is very close to minus 30 degrees. Okay, but in your household freezer, I don't think it goes more than minus 10 degrees Celsius. Okay, very close to minus 5 to minus 10 in your household freezer. Okay, but industrial freezers can go up to minus 30. Then minus frozen products are stored in cold rooms at 18, minus 18 to minus 30 until distributed. Okay, and in order to prevent the growth and spreading. So, what is the principle used here in freezing and chilling? It is usually prevent the growth. Ne? So, it is the second principle, right? Reduce the activity, no? To reduce the activity, right? That is usually the principle. Then we don't usually freeze these. Our fruits, fruit juices, vegetables, right? You don't. So fish and meat usually freeze, but of course, your fruits and vegetables you don't freeze because that's going to dehydrate. Then completely dry them out. No? Okay. So chill at 4 to 7 degrees Celsius. Can you highlight? Freeze at minus 18 to 30. Chill at 4 to 7. You can keep them for a few days, for 10 to 14 days, up to 2 weeks. You can keep if you chill them in the normal. In the fridge, you have two compartments. So there's a freezer and a bottom compartment. Most of it is stuffed into the bottom compartment. That is where the chilling is. Chilling is done. Okay. So we have talked about three food preservation methods. Then we'll move on to salting. Number four, salting. It's very old also, right? Salt has the ability to suck the moisture through the process of osmosis. Yes, because the salt itself is very hydrophilic, what the salt is going to do is when you coat the food in salt, the salt is going to absorb the moisture, then the salt is dissolving and the salt is going to sort of wash away with the water. Okay, so the food inside of the food will be completely dry. So highlight this. Salt has an ability to suck the moisture by osmosis. Because it's a process, it's a diffusion. Why is it diffusion? Because inside the food there is water. In the salt there is a high solute concentration because it's salt. No? So what's the water going to do? The water is going to go from the food to the salt because in order to balance the concentration gradient, because on the salt has of course more solutes. When the food is completely dry from salting, growth is inhibited. Meat is commonly preserved. Can you highlight? You usually salt meat, no? Okay. Bacon is salted and also sort of dried. Okay. What else? Salted. Then of course you have dry fish. Okay. Salted fish. So salting is very commonly closely associated with the drying, no? Because salt is used to dry. It's a way of drying instead of in the sun. So in dry fish, when you make dry fish, salt and sun both. Yes. So what is the principle here? What is the principle? You can take both 1 and 2. Ne? Most likely it is 2. Ne? You reduce the activity. Yeah, you reduce the activity. Right, in preparation. Maybe dried fish. Tuna, dry fish are first salted and then dried in the sun. Yes. Addition of salt make the removal of water fast. Can you highlight this? The salt inhibits microbial growth on the fish, even though some moisture is still remaining. Yes. This is how you make dry fish. Make up Lianba, this question. What are the three principles of food preservation? And the state the role of salt. Taggala within 2 minutes. What are the principles? Yes. Prevention of entry of microorganisms, prevention of their growth and activity, removal or killing of microorganisms in the already in the food. Yes. What is the role of salt in the production of dry fish, particularly in the production of dry fish? What is the role? Yeah, salt makes the removal of water fast and also inhibits the microbial growth. That should be, yes, both things should be written there. So makes the water removal fast also, and inhibits. So this is actually, I mean, because you don't have water, the microbial growth is inhibited. Got it? So it's almost the same thing that we are talking about. Okay, but you had write both points. Got it? Okay. Make a अभी करूँगा इलाका. Right. Tissue culture question we couldn't do. Ne? Okay. Sugar. Sugar is used in syrup form to preserve fruits and in crystallized form to preserve some food items like ginger and orange peel. Okay, right? So these are certain sweets, right? Ginger candy is there, right? Glazed with sugar syrup for the fruits to be to get a superficial protective coating. Some fresh fruits are cooked with sugar and then dried. High sucrose concentration produces osmotic stress on the microorganisms and protects the food from microbial spoilage. Jam, sugar, toffees, ash pumpkin can one puhul dosi kela, have you heard? Right? Not very commonly consumed right now. Okay, ash pumpkin means puhul. Sort of very commercially, it's not a very popular vegetable, but when you make it into candy, it's kind of popular. So, several forms. You can have glazed with sugar syrup as a coating. You can coat certain food items. Okay. Sometimes pineapples are sugar coated like this, I think. Okay. Then fresh fruits are cooked with sugar and then dried. Isn't this about certain blueberries? You can sort of make that's jam, right? That's making jams. So marmalade, I think it's from entire oranges. Marmalade, have you heard? You cook the oranges with the peel in sugar. Then you get like a jam like thing, but it's having a bit of a bitter taste and it's like an orange jam, right? So those kind of sugar toffees, that is again you can make using oranges, I think. Yeah, those kind of things you can do. What was the that question that was there? How does the addition of sugar conserve food? How does the addition of sugar conserve the food? Quickly write this. So we'll finish food preservation today. And then next week we can finish the diseases. And within 2 days, applied biology is done. Okay. Then what is remaining is our sort of final revision topics. You can request if you want a certain topic covered as well. Okay. What's the role of sugar? Yeah. What do you do with sugar? Yeah, the high sucrose concentration produces osmotic stress and protects the food from. So osmotic stress is almost same as the role of salt, right? It's the same as role of salt. You osmotically suck out the water that is going to reduce or inhibit the microbial growth. Okay, few methods are remaining. Let's see. Smoking is the process that cooks, flavors, and preserves food, exposing it to smoke from burning wood. Okay, usually wood smoke is used. Usually wood smoke is used. Smoke is antimicrobial and antioxidant. Most often meats and fish are preserved by smoking. Okay. The aroma generated by the hydrocarbons of burning wood enhances the taste, flavor, and adds smokiness to the meat and fish. So, you know, smoked fish is there and bacon, certain bacons are there, right? So you can have a small, just highlight what are the benefits of the smoke. It is antimicrobial, antioxidant, and also there's a taste and flavor, that sort of a benefit as well. Then you put numbers 1, 2, and 3 to the benefits of smoking. Okay. Then chemicals. Okay. So of course these are the least sort of beneficial, or let's say most harmful type of preservation, and is one of the most commonly used at this during these days. Because why? All the other methods such as drying, smoking, all those things take a lot of time, no, a lot of effort. Now with the use of chemicals, you can do them much more cheaply and much more fast I mean much faster. So they are the widely used types of preservation, but not the best in terms of health. Okay. Chemicals inhibit growth and activity. Okay. And they can kill the microorganisms as well. So what is the principle used here? It could be 2 and 3, né? Both inhibiting the activity as well as the killing of the already living microorganism is done. Preservation of fruit juice. Okay. Production of pickles using chemicals such as sodium benzoate, EDTA, acetic acid, and brine solution. Brine solution is actually it's a salt solution. Acetic acid also, I mean, of course, it's going to reduce the pH. Okay. And yeah, remember these chemicals. Few chemicals: sodium benzoate, EDTA, acetic acid, and brine solution. If you read a food label, you can see most of these chemicals. Okay. Then finally, the sort of the high tech one that is done in sort of advanced labs. Okay. Uh, Limited to radiation from high energy gamma rays. Okay. Highlight the list of waves you can use: gamma rays, X rays, and accelerated electrons. Processing of food by radiation involves the exposure of food into short-wave radiation energy to achieve a specific purpose, such as extension of shelf life, insect disinfection, and elimination of foodborne pathogens and parasites. Okay, so usually this is done as the final process in the food processing. So once you have packeted the food, they usually undergo a final sort of a disinfection process where they are they are subjected to this sort of waves as the final step before they are packaged into boxes and shipped off. Yep. So packets of spices, ground meat, right? That is sort of like the final process of sterilizing them. Okay. So insects, if they are there, they could be destroyed by the gamma rays. Okay. Then certain bacteria and fungi can be there, parasites can be there. Okay. That is like the final uh, protective steps that they take. Okay, right. So we'll stop there for the day. So you can join with the paper discussion this Friday, and we have the rest of the tude to complete. Okay, so we have a lot of upcoming topics such as the filariasis, dengue. Those are the actually the common questions. But today we talked about aquaculture, previously question topic, and then also MCQ value is there in all the propagation methods. Okay. Radiation principle is to prevent entry. No, I told you it is done usually after packaging. So what do you mean? That means it's either to kill. Mostly it is to kill. Okay. The principle of radiation is to kill. Okay. Right. So let's basically. So these are some charts from dengue management. So we'll talk about dengue as well. Alright. So that's all for today. Right. So this Friday the paper class will be happening with the discussion. Okay. So we'll be starting with 2021 paper discussion MCQ. Okay. So please do the paper in your own time. I'll be sending a message to the group as well. 2021 paper. Okay. Adding sugar. Adding sugar is inhibition. Activity, reducing the activity, adding sugar, because you are basically drying out. Okay, you're basically drying out. Okay, as I was saying, this Friday we'll be discussing 2021 paper, okay, MCQ, with sort of detailed reviews into the difficult questions, so that it would be the final revision. Okay, and then we are going to have the guest essay session at the end. Okay, so I'll be sending the two guest essay topics. You have to write before the class and join the discussion. Okay, that's it for the day. Any questions? Okay, you're welcome.